It's a great pleasure to have you again for our 6th episode in JRC1 webinar series. Today we are going to speak about how foods or food substances are modulating our health. Before we go onto the presentation with our invited speaker today, just a few forwards. So I'm Patrick Gonzalez. I will be the moderator today. I'm associate professor at Biotech Paris, and I'm co-coordinator of research and innovation for the UNIgreen University. You can find here my email in case you want to contact us. If you have some suggestion for a webinar that would be interesting for you or anything else, just come get in touch with us. So as a disclaimer, today this event will be recorded as usual and uploaded on the YouTube channel. You can find here the general email address of UNIgreen in case you need a certificate just to certificate that you attended this webinar. You can ask your question in the chat, and I will transfer the question to our speaker today. So about UNIgreen, UNIgreen is an alliance of 8 universities across Europe, and it's supported by the Erasmus Plus program. Our goal within UNIgreen is to push education and research in the field of sustainable agriculture, green biotechnology, and environmental and life science. And so we are going through this goal both through education and through research. We have 8 axes of research within UNIgreen. You can find them here, and today we are doing our webinar in the first axis, which is biochemistry and biotechnology. This is the list of our webinar, and just to mention here, in April we will have a webinar on plant growth promoting microorganisms. So stay in touch to have more information about this coming event. You have also the link here to follow us and also to find the previous videos on our YouTube channel. So coming to today's webinar, so we have the great pleasure to welcome Professor Davide Tagliazucchi. He is associate professor at the University of Modena, and he is a recognized expert in food science, biochemistry, and digestive physiology with more than 100 papers published in this field. And today we will dive into a very interesting topic, which is the relation between phenolic compounds coming from our diet and the relation with colon cancer. So with this, I will let you the stage, Davide. It's really a great pleasure, and we are looking forward your presentation.
Okay, thank you, Patrick, for your kind presentation, and good morning, everyone. I can try to share my presentation. Good morning, everyone, and thank you for being here. Today, I'm going to talk about phenolic compounds and their biological activity, with a particular focus on their preventive role on colon cancer. But what are phenolic compounds? Just to start, phenolic compounds are a large class of molecules characterized by the presence of at least one hydroxyl group directly attached to an aromatic ring. They are plant secondary metabolites widely distributed in the plant kingdom, and we consume them daily through the intake of vegetable foods and beverages. Now, some of them are quite simple. For example, phenolic acids like hydroxybenzoic and hydroxycinnamic acids that contain just one phenolic moiety. Others, like flavonoids, have a more complex structure and contain multiple phenolic rings. In terms of diets, they are found in many common foods, of course, vegetable foods and beverages. They can be found in fruits, like red fruits, berries, or citrus fruits, or fruits of the Rosacea family. They can be found in beverages, like coffee, tea, red wine, or fruit juices. They can be found in vegetables, like onions or eggplants, and of course, in other food, like nuts. Now that we know where they are found, let's see why they are so important for our health. There is strong evidence from epidemiological, observational, and clinical studies that plant-based diet can have important health benefits for us, for humans. Now, today, we know that many of these benefits are related to the presence in these foods of phenolic compounds. In fact, in vitro and in vivo studies have shown that phenolic compounds can help to protect against the onset of several diseases, among others, neurological disorders, cardiovascular diseases, and cancer development and progression. After ingestion, only a small portion of phenolic compounds are absorbed in the small intestine. And after absorption, they undergo phase 2 metabolic reaction. The majority of them, about 80–90%, reach unchanged the colon. Here, they are metabolized by the gut microbiota, producing low molecular weight phenolic acids with high bioavailability and biological activity. These compounds are the so-called gut microbial metabolites of phenolic compounds. These metabolites can then either reach the systemic circulation or can act locally in the gastrointestinal tract and, of course, in the colon. Several of these metabolites have been already identified and shown to be biologically active. However, our knowledge is still incomplete. What is lacking is a comprehensive picture of all the metabolites found after ingestion of phenolic compounds rich vegetable foods. Now let's brief focus on colon cancer. Colon cancer is the third most common cancer worldwide, with nearly 2 million new diagnosed cases and 1 million deaths per year. About 70% of colon cancer cases are attributable to lifestyle factors, with diet, of course, playing an important role both in colon cancer development and progression, but also in colon cancer prevention. For example, diets high in red and processed meat and low in vegetable and fruits are associated to an increase in the risk of development of colon cancer. Whereas, on the other hand, dietary patterns like the Mediterranean diet, which are rich in vegetables, in fruits, and of course in phenolic compounds, are associated to a lower risk of the onset of this disease. The important thing here is that diet is a modifiable risk factor. Therefore, improving healthy diet can in some way prevent the onset of this disease, can help us to prevent the onset of this disease. The interaction with the modulation of gut microbiota is one of the ways in which diet may influence the development and the progression of this disease of colon cancer. In particular, the interaction between phenolic compounds and gut microbiota is of paramount importance. In fact, phenolic compound, as we will see, can shape the microbiota composition towards an eubiosis condition, whereas gut microbiota, gut bacteria, are able to metabolize and modify dietary phenolic compounds, producing these low molecular weight phenolic acids, so these gut microbial metabolites of phenolic compounds. Phenolic compounds can act as anti-cancer agents in different ways, for example, by promoting apoptosis, or by suppressing angiogenesis, or by blocking cell cycle, or by promoting anti-inflammatory condition in our gut. However, until now, the majority of these studies focused on the phenolic compounds present in foods, but now really know that these compounds are totally modified by the gut microbiota. Therefore, the key point here is, can be these gut microbial metabolites of phenolic compounds the real active agent responsible for the protective effect against colon cancer development of a plant-based diet? Based on this, we decided to study the dual interaction between dietary phenolic compounds and the colon microbiota, and consequently their effect on colon cancer. To do this, we try to mimic as close as possible the real physiological condition, so what really happens in our gastrointestinal tract. First of all, we select some vegetable foods rich in specific classes of phenolic compounds, and next we in vitro digested by using an international in vitro gastrointestinal digestion protocol these vegetable foods and perform in vitro colonic fermentation. Next, we analyze the changes in microbiota, the major metabolites and the main metabolic pathways were identified by high resolution mass spectrometry. Then the most important metabolites were tested on 2 colon adenocarcinoma cell lines to evaluate their antiprolifer activity. We selected 2 different cell lines. One is CACO2 and the other one is SW480. Why we selected these 2 different cell lines? We selected CACO2 because this cell line represents a tumor in an early stage of development. So it is important to study the preventive effect of these compounds. On the contrary, SW480 is a cell culture that represents an advanced stage of tumor. They are metastatic, and also they are resistant to common chemotherapeutic drugs. Once assessed the antiprolifer activity, we analyze the cell cycle and the effect of this compound on cell cycle, apoptosis, and reactive oxygen species production. Now we are trying to elucidate the exact mechanism of action of these compounds by applying foodomics techniques, which means metabolomics, transcriptomics, and proteomics. Finally, we are also now studying the possible interaction between this compound and common chemotherapeutic drugs in SW480 resistant cells. We selected some vegetables, fruit and vegetables. For example, oranges, because they are rich in flavonoids, rooibos tea, because it's rich in flavones, blueberry, which is rich in anthocyanins, dark chocolate for flavonols, red onion for flavonols, and eggplants for hydroxycinnamic acids. We perform in vitro digestion and colonic fermentation, and then we analyze the changes in the microbiota. You can see here the data about the changes in some beneficial bacteria taxa and opportunistic bacteria taxa after digestion of eggplants and in vitro fermentation of eggplants, which is rich in hydroxycinnamic acids, compared to a control digestion without vegetable foods. As you can see in the slide, hydroxycinnamic acid were able to foster the growth of beneficial bacteria taxa, and at the same time, decrease in the growth of opportunistic bacteria taxa. In particular, hydroxycinnamic acid were able to promote the growth of Faecalibacterium prausnitzii, Akkermansia mucinifila, Clostridium 4 group bacteria, and Lactobacillus. All these bacteria are associated to a lower risk of development of colon cancer. On the other way, hydroxycinnamic acids were able to inhibit the growth of some opportunistic bacteria like Escherichia coli or Enterobacteriaceae. These bacteria are related to the promotion of intestinal inflammatory condition, which are associated to an increased risk in developing colon cancer. Similar data were then obtained when we digested red onion rich in flavonols, with always an increase and a promotion of the growth of beneficial bacteria taxa, and vice versa, a decrease in the growth of opportunistic bacteria taxa. Similar results were also obtained when we digested blueberries rich in anthocyanins, dark chocolate rich in flavonols, oranges rich in flavonols, and rooibos tea rich in flavones. Of course, there are some differences between a different food, and especially dark chocolate was less effective than the other vegetable foods, but probably because this is a complex matrix with other components, other compounds that may have an effect, an impact, influence our gut microbiota. Overall, these results suggest that phenolic compounds found in food can modulate our gut microbiota towards aneubiosis and protective condition, and therefore protective towards the onset of colon cancer. This protective effect is related to the promotion of the growth of several beneficial bacteria which are involved in decreasing intestinal inflammation and in decreasing the risk of developing colon cancer. For example, Akkermansia mucinifila and Clostridium 4 group bacteria are able to inhibit the release of inflammatory factor from immune cells, potentially decreasing intestinal inflammation. Bacteroides and Fecalibacterium prausnitzii are short-chain fatty acids producing bacteria, and we know that short-chain fatty acids are important in the prevention of colon cancer. Finally, Lactobacillales are probiotic bacteria involved in enhancing intestinal barrier function and improving intestinal barrier integrity. By promoting the growth of these bacteria, phenolic compounds present in our vegetable foods are able to reduce potentially in the colon the intestinal inflammatory condition and therefore protecting against the onset of colon cancer. Next, we elucidate the different metabolic pathway for the microbial metabolism of the different phenolic compounds. Here you can see the data about hydroxycinnamic acids. The first step in the microbial metabolism of hydroxycinnamic acid is ester hydrolysis, which with the release of the different agglicons. Here you can see one agglicon just as an example, which is dihydroxycinnamic acid. Next, these agglicons can be subjected to further metabolic reaction like reduction, dehydroxylation, beta or alpha oxidation, which resulted in the formation of the gut microbial metabolites of hydroxycinnamic acids that are low molecular weight phenolic acid like hydroxyphenyl propanoic acids, hydroxyphenyl acetic acids, and hydroxybenzoic acids. The most important metabolite found after the fermentation of hydroxycinnamic acid was free hydroxyphenyl propanoic acid that is circled in red. The metabolism of flavonols started with the deglycosylation of glycosylated flavonols, releasing the agglicons like quercetin and isoramnitin that are the most important flavonols agglicons. The agglicons are then subjected to further metabolic reaction. The first one is the serine fission with the opening of the serine as reported here in the figure. The serine fission produce intermediate metabolites that are further metabolized always by beta oxidation, alpha oxidation, or dehydroxylation reaction to generate always hydroxyphenyl propanoic acids, hydroxyphenyl acetic acids, and hydroxybenzoic acids, the same metabolites found after fermentation of hydroxycinnamic acids. The metabolism of flavanols started always with the deglycosylation of the glycosylated flavanols, which resulted in the release of the flavanone agglicons, in this case asparagine, eryodictyl, and naringenin. We found that these compounds accumulated during in vitro colonic fermentation, but they are also, despite their accumulation, subjected to further metabolic reaction. The first step is serine fission, which can be carried out by two different pathways, as reported in the figure. Independently on the pathway, the final result of the metabolism are always hydroxyphenyl propanoic acids, hydroxyphenyl acetic acids, and hydroxybenzoic acids. The metabolism of flavones converged on that of flavanols. The first step is always the deglycosylation of glycosylated flavons, releasing the agglicons. In this case, the flavone agglicons were apigenin, luteolin, and methylluteolin. These agglicons are next subjected to reduction, so mainly dehydrogenation, producing the flavanone agglicon, asparagine, eryodictyl, and naringenin. Then the metabolism followed the same pathway as observed before, we always observed the accumulation of aglycons and production of low molecular weight phenolic acids. The metabolism of flavanols was quite different. The first step was the hydrolysis of oligomeric flavanols that results in the release of the flavanol aglycons like catechin, as shown in figure, but also epicatechin and others. These compounds can accumulate during in vitro colonic fermentation, but they are anyway subjected to further metabolism. The metabolism of flavanols is a little bit different because started with the C ring opening, as reported here, followed by A ring fission. This fission produced different metabolites, which are hydroxyphenylvalerolactones and hydroxyphenylvaleric acids. These metabolites accumulate during in vitro colonic fermentation, but they are also further metabolites, always generating hydroxyphenylpropanoic acid, hydroxyphenylacetic acids, and hydroxybenzoic acids. The metabolism of anthocyanins followed two different pathways. In the first one started always with deglycosylation, releasing the aglycons, like as an example, delphinidine and chanidine. This underwent C-ring fission, as reported in the figure, generating an unstable chalcons that underwent hydrolysis, releasing the corresponding hydroxybenzoic acids, like 3-hydroxybenzoic acid, which is also known as gallic acid, or dihydroxybenzoic acid, which is also known as protocatechoic acid. The final product of anthocyanin metabolism was pyrogallol, which is this benzene 1,2,3 triol. The second pathway converged in the pathway of flavanols after the glycosylation. In this case, the aglycons from anthocyanins were dehydrogenated, generating the flavanols aglycons that can accumulate during colonic fermentation, but also they were subjected to further metabolism, generating hydroxyphenylvalerolactones, hydroxyphenylvaleric acids, and hydroxyphenylpropanoic acids, hydroxyphenylacetic acids, and hydroxybenzoic acids. So now, after this study, we know that really the microbial metabolism of dietary phenolic compounds converged in the formation in few key metabolites independently on the class of phenolic compounds. We selected these metabolites for further cell culture experiments, and you can see the selected metabolites in the table. We selected monohydroxylated and dehydroxylated phenylacetic acids, monohydroxylated and dehydroxylated phenylpropanoic acids, mono and dehydroxylated phenylvalerolactones, some hydroxybenzoic acids, the most important, which are protocatechoic acid, which is dihydroxybenzoic acid, vanillic acid, that is hydroxymethoxybenzoic acid, and gallic acid, that is free hydroxybenzoic acid. We also selected pyrogallol, which is the final metabolites found in the pathway of anthocyanins, and also we selected the aglycons that accumulates during fermentation, during microbiota metabolism, which are epicatechin and catechin, that are the flavanol aglycons accumulating during the microbial metabolism of flavanols and anthocyanins. We selected also naringenin, eriodictyl, and asparagine, which are the flavanol aglycons that accumulates during the microbial metabolism of flavanols and flavons. Next, we tested this metabolite on the two different coronal adenocarcinoma cell lines to test their anti-proliferative activity. You can see here the data about CACO2. 5 compounds were active already after 24 hours of incubation of CACO2 with these compounds, and presented EC50 value between 25 and 50 micromoles per liter. The EC50 value decreased as the incubation time increased, reaching the lowest value after 72 hours of incubation, with EC50 value around 50 micromoles per liter for the most active compounds that are dihydroxyphenylacetic acid, dihydroxyphenylpropanoic acid, dihydroxyphenylvalerolactose, and gallic acid. Pyrogallol show a little bit higher EC50 value, about around 30 micromoles per liter. There were additional compounds active, like the aglycons epicatechin and catechin, that showed EC50 value between 50 and 80 micromoles per liter after 72 hours of incubation, and protocatechoic acid, which show EC50 value around 100 micromole per liters after 72 hours of incubation. Regarding SW480, only 5 gut microbial metabolites were found to be active. The most active were the aglycons naringenin, eriodictyl, and asparagine, coming from flavanones and flavones metabolism. They presented the EC50 value ranging from 50 to 80 micromoles per liter after 72 hours of incubation. As you can see, also gallic acid and pyrogallol were active, although less than the flavanones aglycons. Regarding cell cycle analysis, we found that, in this case, we are talking about CaCO2, we found that 2 compounds, dihydroxyphenylacetic acid and dihydroxyphenylpropanoic acid, were able to block cell cycle in G2M phase, whereas dihydroxyphenylvalorolactone was ineffective. 2 additional compounds, gallic acid and pyrogallol, induced apoptosis in CaCO2 cells, and the apoptosis induction was confirmed by analyzing the caspase-free 7 activation, as you can see in the graph on your right. All the other compounds were ineffective in blocking or altering the cell cycle. Anyway, these compounds were able to promote the production of reactive oxygen species in CaCO2 cell by promoting a condition of oxidative stress, explaining their antiprolifer activity observed during incubation with CaCO2. In the case of SW480, you can see that 3 compounds, pyrogallol and 2 aglycons, eriodictyl and asparagine, were able to block the cell cycle in G2M phase, whereas gallic acid and naringenin were ineffective. No apoptosis induction and no reactive oxygen species productions were found in SW480. Now we are trying to elucidate the exact mechanism of action of these compounds by applying foodomic techniques, which means the application of metabolomics, proteomics, and transcriptomics on cultured cells after the incubation with our metabolites that were found to be active in the first set of experiments. I can show you just some data about metabolomics, because transcriptomics and proteomics are still ongoing. In particular, I want to show you some data about the incubation of some compounds with SW480. Here you can see the data about the incubation of the SW480 with gallic acid. Of course, are metabolomics data, so we analyze the metabolome in cells exposed to gallic acid and compare them with control cell not exposed to any compounds or drugs. And the most important result we found was a downregulation in the level of phosphocholine and an up-regulation in the level of glycerophosphocholine after incubation with gallic acid, which resulted in a downregulation in the synthesis of several phospholipids, including phosphatidylcholine. Here on the left, you can see the phosphatidylcholine biosynthetic pathway. They start with the production of choline, which is then phosphorylated by choline kinase in phosphocholine, and then is included in phospholipids to generate phosphatidylcholine. Our hypothesis based on this data is that gallic acid downregulate some enzymes in the phosphatidylcholine biosynthetic pathway, in particular choline kinase, resulting in a decrease in the level of phosphocholine. And another enzyme which can be downregulated is glycerophosphodiesterase phosphodiesterase 6. The downregulation of this enzyme results in an accumulation of glycerophosphocholine. Therefore, our hypothesis that downregulated this enzyme, gallic acid, may decrease or block phosphatidylcholine synthesis, resulting in a block in the cell proliferation. This effect has been already seen with other drugs in other cell lines. For example, doxorubicin, which is a chemotherapic drug, is able to block the proliferation of breast cancer cells by perturbing the biosynthetic pathway of phosphatidylcholine, exactly as observed in our experiment with gallic acid. In addition, we found an upregulation of some metabolites of leucine catabolism, which are free methylglutaric acid, N-isovaleryl glycine, and free hydroxyisovaleryl carnitine. Leucine is one important amino acid for SW480 because it is catabolized to produce acetyl-CoA and acetoacetate that fuel the Krebs cycle inside the mitochondria, which is the most important pathway for energy production in SW480. The metabolites that we found upregulated were really secondary, were metabolites coming from the secondary catabolism of leucine. The primary catabolism of leucine, as already told you, produces acetyl-CoA and acetoacetate. The secondary catabolism produce these metabolites. The upregulation of these metabolites is important because we hypothesize that gallic acid is able to downregulate leucine catabolic enzyme, promoting the flow of metabolites in the secondary catabolism. In this way, decreasing energy supply to the SW480 cells, starving the cancer cell and blocking cell proliferation. I want to show you just the last data, then I finished my presentation, about the effect of aspartame and erydicyl to aglycon on SW480. These are always data from metabolomics experiment. The two aglycons act in the same way, but they are strictly structurally similar. The first, the most important data that we found after metabolomics experiment was an accumulation in cells of several amino acids, including glutamate, branched chain amino acids, proline, methionine, and so on. As already told you before, these amino acids are widely used by SW480 to get energy. In fact, in these cell lines, this cell line get energy, especially from mitochondrial catabolism, including beta oxidation of fatty acids, catabolism of amino acids, and of course, Krebs cycle and oxidative phosphorylation. We hypothesize that the accumulation of these amino acids is due to an induced mitochondrial damages by aspartame and erydicyl. The induced mitochondrial damages, of course, cause a decrease in energy production by mitochondria, and which result in a starving of cancer cell in a block of proliferation. This idea is supported also by additional data. For example, we also find an accumulation of citric acid, which means alteration in Krebs cycle, which is always inside the mitochondria. But also we found an accumulation of several intermediates of the glycolytic pathway. That means an increased flux of metabolites in the glycolysis. So the hypothesis that cells try to switch the energetic metabolism from a mitochondrial energetic metabolism to a glycolytic energetic metabolism to try to supply to the lower energy production after damages to the mitochondria. Moreover, we found in literature that some drugs used as chemotherapeutic agents are able to induce mitochondrial damages in cancer cells, and often these mitochondrial damages are associated with a block in G to M phase of the cell cycle, exactly as we found for SW480 as shown you before. Finally, we also found upregulation in the level of some metabolites of the purine catabolic pathway, like EMP, GMP, guanosine, inosine, hypoxanthine, guanine, and xanthine. And this is coherent with cell cycle blocking because there is blocking the cell cycle means reduction in DNA synthesis, which resulted in an accumulation of nucleotides, which are pushed towards the catabolic pathway. Moreover, we also found an upregulation of two intermediate of the NAD catabolic pathway, like nicotinamide and 1-methyl-nicotinamide. Also, this is coherent with the hypothesized mitochondrial damages because the majority of NADH is produced inside the mitochondria. Lowering this production may result in an increase in ADA and NAD concentration in the cells, and of course, accumulation of NAD increase the flux of metabolites towards its catabolic pathway. To conclude, foods rich in phenolic compounds can promote anti-inflammatory, anti-cancer condition in the gut. Chronic fermentation of phenolic compounds converge in the formation of few key metabolites that enhance intestinal abiosis, promoting beneficial bacteria growing and limiting the growth of opportunistic ones, potentially reducing intestinal inflammation and therefore protecting against the onset of colon cancer. But not only, most of the colonic metabolites show anti-proliferation activity on the two colon carcinoma cell line tested, especially on the CACO2 cell line, which is important from a preventive point of view. The mechanism of action of this compound have been identified. Some block or slow the cell cycle, some induce apoptosis, some promote oxidative stress, and the studies are still ongoing. Finally, what we can say that a diet rich in phenolic compounds can modulate the gut microbiota and lead to the production of microbial metabolites with anti-cancer activity, potentially explaining the preventive effect of a plant-based diet on colon cancer development. I want to thanks all my collaborators at the Nutritional Biochemistry Lab at the University of Modena, Reggio Emilia, and of course, thank you for your kind attention. Thank you.
Thank you very much, Davide, for this presentation. I think that Hippocrates would be amazed seeing this result.
Of course.
Since he was thinking that food would be our medicine, right? So yes. Here we have a really strong rationale for this statement.
Yes, of course. And not only for colon cancer, but also for more diseases, like there are strong evidence for the protective role in cardiovascular diseases.
Mm-hmm. Yeah.
Both from epidemiological, but also clinical, in vitro studies, etc., etc. So okay. I think that diet is really important for our health.
Yeah, this is the starting point where we have
yes, because we are continuously introducing molecules with the diet. Okay, some are nutrients, so are considered endogenous by the organism, but most of the other molecules are exogenous, so they can be protective as phenolic compounds, but also they can promote the development of disease. So,
yeah,
and
sure.
In the prevention of disease, diet is really important and play a really important role.
Yeah, sure. So if I can summarize a bit what I saw from what you said. So there are some direct and indirect effects of the polyphenol, right? Some direct effects on cancer cell, for example, and indirect effects on immune system, but probably also on other cells in the gut.
Yes.
You showed that there are some effects on Akkermansia, for instance. And Akkermansia has been shown to increase the thickness of the gut barrier. Is this something that you observed?
Yes, we found more or less all the phenolic classes promote the growth of Akkermansia mucinifila in the gut. So, and yes, it is linked to a protective effect against the development of colon cancer, both by improving the intestinal barrier, but also by inhibiting the release of inflammatory factors from immune cells. So,
yeah, and there are also some effects on on diabetes, for example, or obesity. These have been shown in literature.
Yes, also on obesity and of course diabetes, which is strictly linked to obesity. And yes, they may have an effect of also in obesity, and they are studied for the anti-obesity effect. So just to it's not only a matter of calories, okay, but also of metabolism.
Yeah. I was very surprised by the effect of polyphenol on good bacteria and bad bacteria. It seems that it's black and white. You have stimulation of the growth of good bacteria, and conversely, the bad bacteria are reduced in abundance. What does it tell us, actually? Is there a specific pathway in metabolic pathway in bad bacteria that make them bad, or?
Really, I don't know, because I'm not a microbiologist, but a biochemist. What I know is that the phenolic compounds, as found in foods, are antimicrobial. So normally they inhibit the growth of bacteria. But now we are talking about a co-evolution between the phenolic compounds and gut bacteria. So probably, okay, we eat vegetable food, so phenolic compounds from thousands of years. So just when we were also the ancient Homo sapiens just eat vegetables. So, and the idea is that there was a co-evolution between the gut bacteria and phenolic compounds. So phenolic compounds select right bacteria in the intestine, only bacteria able to metabolize phenolic compounds, which are the beneficial bacteria. These bacteria are able to take phenolic compounds, metabolize it, and making it inoffensive for them. Okay, so they can grow and have beneficial effect. And for opportunistic bacteria, maybe is prevalent the antimicrobial effect. So,
so it's a matter of co-evolution in a way.
Yes, it's a matter of co-evolution. So it is just a very, very new argument that we are exploring in the last conferences about phenolic compounds. But is a new idea, just to try to explain this effect.
That's exciting. So meanwhile, Davide, I have also a question about the mechanistic effects on cell lines. You can have different type of cell deaths, right? And you went for apoptosis. Did you try to look to other kind of cell deaths?
Not yet. We didn't check for necrosis of autophagy. Yeah, but we are working on this.
I was thinking about this because 50% of colon cancer have a deficiency in BAX, which is a main driver of apoptosis. And so I don't know what's the status of BAX in CACO or SW480, but maybe they are not doing apoptosis, but they can die through other type of pathways.
Yes, of course. We other pathways like autophagy or necrosis, but also by blocking the cell cycle and not killing really the cells, but just blocking their proliferation. Okay, or as we hypothesize for gallic acid, just by blocking the biosynthesis of phosphatidylcholine, so cells are not more able to divide and proliferate. So it's not only a matter of killing cell, but just blocking also the proliferation.
Okay, so in SW480, you mentioned you don't have apoptosis, but you don't see any death of the cells? It's just cytostatic, or do you see some cells dying?
No, we see some cells dying, but in the majority of the case, they are really static, so they stop to grow.
Okay.
grow, to divide, to proliferate. And of course, I think that the diet cells are a consequence of the incubation time, so they need to divide continuously. If not, they start to die. So, but we didn't see any apoptosis effect, and I think also autophagy and necrosis are not present. But we are working on this, especially in SW480.
Yeah, because it would be very interesting to see if these compounds can bypass resistance to apoptosis and shift cell death towards other kind of cell death.
Yes, for sure, SW480 are resistant to apoptosis, I know. And also they are resistant to apoptotic chemotherapeutic drugs. So,
okay, yeah,
we will see in next seminars.
Yeah, hopefully. Related to the point that you made about phosphatidylcholine, I was wondering whether you saw also an increase of ceramide.
No, we didn't find an increase of ceramide.
Okay. Because
yeah,
this was also the point about alternative cell death, because you can have, for example, if ceramide accumulates, you can have instability in lysosomal membranes, release of lysosomal enzyme, and this can trigger cell death.
Yes.
More like a necrosis effect, but
yes, there are some drugs that increase the ceramide levels, but we didn't find an increase in ceramide. But also, we are talking about only metabolomics data, so we need to. We have this data about the changes in the metabolome after incubation with these compounds, but we have to confirm our hypothesis by applying transcriptomics and proteomics data to see if really there is a downregulation or upregulation of some enzyme and in the different metabolic pathways.
Yeah, I understand. You also mentioned, there is a question by Uriel. Did you study the microbiologic impact of phenolic compounds on the food matrix and the impact on the human system?
No, we didn't. We didn't study the effect of phenolic compounds on the bacteria present in foods. So I know that there are some research groups that are trying to use it as a preservative during food conservation, like in packaging or something similar, because they are antimicrobial. So I think that they are able to block the growth of pathogenic bacteria in foods, but we didn't study this aspect of phenolic compounds.
Okay, so Dr. Elal wanted to send his regard to you.
Hi, Ahmed.
About the oxidative stress you mentioned, do you have an idea about from where it's coming? You mentioned that there are some alteration of mitochondria. Is this the main source of ROS in your system?
Okay, we really don't know this, but I think in CACO2 cells, the source of reactive oxygen species are the phenolic compounds itself. We found that the compounds able to increase the ROS production are compounds which contain a catechol group, which means an aromatic ring with 2 hydroxyl group, and this is prone to oxidation. So we think that the presence of oxygen causes an oxidation of the catechol group of phenolic compounds and increasing the production of reactive oxygen species. So promoting oxidative condition in the cell and increasing the reactive oxygen species inside the cell.
Okay.
In CACO2. In SW480, we hypothesized mitochondrial damages, but we didn't find any increase in reactive oxygen species. There is also quite some differences between the two cells, because CACO2 cells are slowly proliferating cells, and they also use glycolytic pathway for production of energy, ATP, while SW480 are highly proliferating cells, which are not in anaerobic, but live in an hypoxic world. and they use oxygen very, very fast through the mitochondrial energetic metabolism. So maybe this is the point why we found ROS in CACO2 and not in SW480.
So they are relying much more on mitochondria. And so when you are treating these cells with SP18, you mentioned that there are a lot of damaged mitochondria, and ultimately maybe the cell dies because it doesn't have enough energy to support survival. Have you looked at mitophagy? Because if you have some alteration of mitochondria, probably you have some autophagy trying to remove these mitochondria, and this can lead to a deficiency in mitochondria, which can then trigger cell death.
Yes, okay. This is a very good point, and I think that we will try to look for this effect on mitochondria, because at the moment we didn't study this. We have not studied this aspect, but could be is an interesting idea.
Yeah, could be. It might be that you exhaust the cell from mitochondria because you trigger mitophagy with your compound.
Yes, could be, because we hypothesize mitochondrial damages, but we don't know how this compound can damage mitochondria. Maybe is related to this effect. So we will wait for transcriptomic and proteomic results, then we can try to go more in deep in the mechanism.
I have a more general question. So you showed all these effects, and they are related to microbiome composition. So do you think that every person reacts differently to these compounds?
Yeah.
Do we have to have a specific signature to have the good effects? And did you identify the signature?
Yes, of course. We talk about metabotypes. Because microbiota is different person by person. So, and in the last 10 years, a lot of studies have been carried out to understand the metabolism by microbiota of phenolic compounds. And what some researchers found is that some volunteers are able to metabolize this, and you can find these low molecular weight phenolic acids, these metabolites, in your body, in your plasma, in your urine, and others are not able to metabolize because they don't have the exact specific bacteria able to metabolize it. For example, there are a lot of studies about green tea, because the flavonols have metabolism a little bit different from the other phenolic compounds. They need specific bacteria to cleave the A ring, whereas all the other phenolic compounds are metabolized by cleaving the C ring. And this is carried out only by specific bacteria taxa, okay? So when this group started to study the metabolism of flavonols and the bioavailability of flavonols, they found that in some people the bioavailability was like 10-20%, in other was 100%, so totally depending on the microbiota. We don't know which microbiota we have.
We have to go towards personalized.
Yes. This is the point. We have
advice for dietary, which is
We have to move towards personalized nutrition, and just both for the prevention of disease, but also for the management of the disease. In fact, we are trying to. We are now studying. I didn't show you the data because if not, it was too long the presentation. But we are studying the combination of our compound with chemotherapy drug in resistant SW480, with the scope to arrive to have a personalized nutrition. So if we know now we have the first data that these compounds are able to enhance the effect of fluorouracil, which is one of the most important chemotherapies used in colon cancer. And they are able also to decrease the AC50 of more than 10-15 times. and this is going in this direction. So if we know which metabolites are able to do this effect, also we can understand which type of nutrition is better for oncologic patient taking this medication. Okay. Because also here you don't know if your cancer cells are resistant or not. And often they became resistant after a different cycle of chemotherapy. So this is an important point.
Yeah, this is beautiful biology in a way that it's integrative. It's not on one point, it's really integrating many aspects. Okay. Thank you very much, Davide. I don't see any comment or question in the chat, so I would like to thank you again for your presentation.
Okay, thank you too, and thank you for the opportunity to present my work in UNIgreen webinar.
Really a pleasure, and I guess our community will also appreciate all the data that you show and the ideas also. Thank you very much.
Thank you very much.
Bye-bye. Bye.